Today we're going to be learning about Gregor Mendel, an Austrian scientist who's considered the father of modern genetics thanks to his experiments with pea plants. We're going to look at some of his experiments and talk about some of his findings. Here's a picture of Mendel. So he was an Austrian monk and lived on a monastery in the 1860s. And he worked mostly in the monastery garden and did experiments on the plants in that garden. Those experiments yielded some very interesting results, and thanks to his work, Mendel is considered the father of modern genetics. The plants that were growing in the monastery garden were pea plants, and the pea plants differed from each other a little bit, and Mendel looked at 7 characteristics shown in this table of that distinguished the pea plants from each other. So some were tall, others were short. Some had round seeds, others had wrinkled seeds. Some had yellow seeds, others had green seeds, etc. So Mendel observed and recorded characteristics of these pea plants. Mendel studied the peas over several generations. So he came up with a very clever way of crossing the peas with each other. And he looked at the peas in the parent generation, and then he would cross plants from the parent generation, and that the offspring of that cross he called the F1 generation. So if you think of the P generation as the parents, the F1 generation is like the children. And then he took those plants and crossed them together. The offspring of that cross he called the F2 generation, which you could think of as being sort of like the grandchildren. So he looked at the parent generation, the children of those parents, and then the grandchildren. So we studied 3 generations of peas. Pea plants were an ideal plant for Mendel to study because they reproduce really fast. So he could look at a bunch of generations in a very short amount of time. And it's very easy to control which peas mate with which others. So basically what he did is he used a paintbrush, and he would take pollen from one plant and dust it on the other. And that way he was able to cross 2 parent plants together and know which parents gave rise to a certain offspring. So he was able to say, okay, I have this plant, plant A and plant B, and he was able to cross them together and get plant C, and know that A and B were the parents of C. The first experiment Mendel did was called a monohybrid cross. So he took 2 peas that had one characteristic that was different, and he crossed them together. So for example, here's an example of a monohybrid cross involving flower color. So here we see a monohybrid cross between a purple and a white flower. So Mendel would cross the parents together. He would observe the characteristics of the children, and he would then cross those children and look at the characteristics of their offspring. So that was called a monohybrid cross. And he got this really interesting result, which we can see here. So if you cross a purple and a white flower, you get all purple flowers for the offspring. If you then take the offspring and cross them and look at the grandchildren, most of them will be purple, but some will be white. So the children of a purple and white are purple, but the grandchildren, we see some of the grandchildren are white. So it's an interesting result. And this led Mendel to his first major finding, which wound up to be correct. His first major finding was that the reason that different pea plants had different characteristics is because there's alternative forms of genes called alleles, which can lead to those different characteristics. So for example, for flower color, you could have a purple allele or a white allele. So there's alternative forms of genes called alleles. If we remember from last week, a gene is a specific region of DNA that codes for a specific protein. If you have a different allele for a certain gene, you'll code for a slightly different protein, and you'll have a slightly different trait than somebody with a different allele than you. So in humans, we have 23 pairs of homologous chromosomes, and the homologous chromosomes, they generally have the same genes on them, but the different chromosomes might have different alleles. So for example, if we looked at the first pair, both of these chromosomes might have an allele for eye color, but the one on the left might have the brown eyes allele, and the one on the right might have the blue eyes allele. So they have the same gene, which is eye color, but they might have different versions of that gene. The second thing Mendel wound up finding is that for each characteristic, an organism inherits one allele from each parent. These alleles may be the same or they may be different. If an offspring has two of the same allele, we say it's homozygous. If it has two different alleles, we would say it's heterozygous. We write if an offspring is homozygous, we would write its alleles like this, using either two capital letters or two lowercase. That represents that offspring has two of the same allele for that gene. If the offspring has two different alleles, we would write it as one uppercase letter and one lowercase. So that would mean that the offspring has two different alleles for that particular gene. This also relates to Mendel's law of segregation, which states that an organism's two alleles for each gene segregate during meiosis. That means that when the parents breed with each other, their alleles separate, and mom is going to pass down one allele and dad will pass down one allele, and then the offspring will get its two alleles that way. So mom's alleles separate, dad's alleles separate, and then two of them at random are going to combine with each other, and that's how you get the two alleles of the offspring. So if we remember how sexual reproduction works, the sex cells or gametes from each parent are going to fuse together to make a zygote. The zygote then divides a bunch of times and then grows into a fully developed organism like you or me. So each sex cell has one allele for each gene in it. And when we put the sex cells together, we get a zygote that has two alleles for every gene. So each individual is going to get one allele for each gene from mom and one allele from each gene from dad. And then you'll have the full two alleles per gene. Here's a little visualization of meiosis, just to review. And we can see that the homologous chromosomes separate. So the homologous chromosomes separate, and then here's our gametes. Each of these chromosomes has one allele per each gene on it, and then two of these sex cells will come together to make a zygote that has a full set of alleles for each gene. So we can see the homologous chromosomes, they separate, and then they wind up in the gametes, and then those gametes are going to fuse into a zygote that's going to have a full set of genetic material. So what if you have two alleles for a gene, but they're different? So if you have like a brown eyes allele from mom and a blue eyes allele from dad, are you going to have brown eyes or blue eyes? So in general, if you have two different alleles for a gene, one of those alleles will be what we call dominant, So when two alleles are present, but only one of them is expressed, we say that allele is dominant over the other. So in the case of brown eyes and blue eyes, brown eyes is the dominant allele. So if you inherit brown eyes from dad and blue eyes from mom, you will have blue eyes you'll have brown eyes because brown is considered dominant. And for most genes, one of the two alleles is going to be dominant. So in the bottom right here, we see an example where round is dominant to wrinkled. Round is represented with a capital S and wrinkled is represented with a lowercase s. We can see that each P has two alleles for each gene like we would expect. If we have two big S's, so two round alleles, the seed will be round. If we have two little S's, so two wrinkled alleles, the seed will be wrinkled. If we have one of each allele, so one round and one wrinkled allele, then the seed is going to be round because the round allele dominates the wrinkled allele. By the way, the allele that's not dominant is considered recessive. So the dominant allele is expressed when there's one of each, and the recessive allele is the allele that's masked when there's one of each allele. Two more quick vocab words, and then we're all set with today's notes. So the alleles an organism has for a specific gene is called the organism's genotype. And for each gene, we represent the alleles by using two letters. We use a capital letter for the dominant allele and a lowercase letter for the recessive allele. So the two alleles an organism has for a given gene, we call the organism's genotype. If the organism has two of the same allele, we say the organism is homozygous for the gene. If it has two different alleles, we say it's heterozygous for that gene. So what alleles an organism has is its genotype. Phenotype is the characteristics an organism has as a result of their genotype and environment. So anything we can observe about an organism is its phenotype. So if I say, so Mr. Rosen has brown eyes would be an example of phenotype. Mr. Rosen has a dominant and recessive allele for eye color would be considered genotype. Okay, we're all set for today's notes.